Thanks for joining us in another fascinating journey through the world of science here on the Sands Channel podcast.
Great to be here.
If you're finding these explorations interesting, please do take a moment to like this, and subscribe. Your support really helps us bring you more of these deep dives.
It really does.
Today, we're looking at something truly fundamental in biology. How even seemingly tiny alterations within our genetic code can create a cascade of effects, reshaping the entire landscape of gene activity within our cells.
Yeah, it's like the ripple effect, right?
Exactly. Think of it as understanding how a small change in one part of a complex machine can have widespread consequences for how the whole thing operates.
That's a great way to put it. We're essentially investigating the interconnectedness of the cell at its, most basic level.
And the insights we're diving into today come from a really interesting study using some pretty advanced techniques.
That's right. A compelling study in Nature Genetics that used some cutting-edge methods to really probe this question.
Okay, let's dive in then. The first technique you mentioned, Perturb-seq. Sounds sophisticated. Can you tell us a bit about what that lets researchers do?
Absolutely. So imagine being able to make very precise edits to thousands, literally thousands of different genes in a population of cells.
Okay, thousands.
And then for each individual cell in that whole population, you can get a really comprehensive snapshot of which genes are active, which aren't.
Wow, okay. Per cell.
Per cell, that's the power of Perturb-seq. It cleverly combines CRISPR gene editing, the tool for making those precise genetic changes.
Right, CRISPR.
With single-cell RNA sequencing. And that lets us see what each individual cell is doing with its genes instead of just getting an average from the whole group.
So you can see the specific effects of, say, changing gene A in this cell and gene B in that cell.
Exactly. It allows scientists to observe the effects of a huge number of genetic changes simultaneously.
That's an incredible amount of data. But I imagine, as the paper notes, analyzing that kind of data must have its own challenges, like noise or maybe missing smaller effects.
You're spot on. While Perturb-seq is super scalable, the sheer volume of data, it inevitably contains some random variation noise, right? Mm-hmm. Which can sometimes mask the real biological signals. Mm-hmm. Plus, because you can only analyze, a limited number of cells for each specific genetic tweak.
Right.
Very small, but maybe real effects on gene activity might be hard to reliably pick up with standard methods.
Okay, so how are researchers getting around that to get a clearer picture? That brings us to this new method, TRADE. Is that right?
Exactly, TRADE. It stands for transcriptome-wide analysis of differential expression.
Okay.
It's basically a new statistical approach developed specifically to handle this kind of complex data from things like Perturb-seq.
And what's the key idea behind it? How does it overcome those hurdles like noise and missed effects?
Well, the core idea is, instead of just focusing on whether individual genes show a statistically significant change after you tweak another gene.
Right, like the standard approach.
Yeah. TRADE tries to model the overall distribution of the true gene expression changes across the entire transcriptome, all the active genes in the cell.
So it's looking at the bigger picture, the whole landscape, not just the tallest trees that fell.
Precisely. It takes into account the measurement errors that are always there and estimates something they call the transcriptome-wide impact, or TI.
TI. Transcriptome-wide impact.
Yeah. Think of TI as a score. It tells us the overall disturbance, the total effect caused by that one genetic change across all the genes.
So back to your ripple analogy. It's measuring the size of the whole ripple, not just the biggest waves.
That's a perfect way to put it. Traditional methods might focus on the genes most dramatically up or down regulated. TRADE tries to quantify the total impact, including all those smaller, subtler shifts across the board.
That makes a lot of sense. So when the researchers applied TRADE to the Perturb-seq data, what kind of key findings emerged? What did they learn about these ripple effects?
Well, one really striking thing was just the sheer scale of these effects. They estimated that a sort of typical genetic change actually influences the activity of about 45 other genes.
45. Okay, that's quite a few.
It is. But here's where it gets really interesting. When they looked specifically at essential genes, the ones absolutely crucial for a cell to survive.
Yeah, the critical ones.
Right. A typical tweak to one of those could impact over 500 other genes.
500? Seriously?
Over 500. It just really underscores how incredibly interconnected these cellular systems are. You disrupt one key player, especially an essential one, and the effects spread really far.
Wow, that has huge implications if you're thinking about, say, drug targets. Messing with an essential gene might have really broad, maybe unintended side effect.
Absolutely. That's a key consideration.
So that 500 number really hits home how central those essential genes are. Now, did the study also look at whether these effects are the same in different kinds of cells? Like, is a liver cell affected the same way as a nerve cell?
They did look into that, and while there were some, general patterns, they definitely found significant differences depending on the cell type, which makes sense, right? Different cells have different jobs.
Sure.
So, for instance, changing the GATA1 gene had a particularly strong effect in K562 cells. Those are kind of like precursors to red blood cells.
Okay.
And GATA1 is a known master regulator for making red blood cells, so that fits perfectly.
Right, context matters.
Exactly.
Yeah.
And similarly, messing with a gene called HMGCR showed strong effects in Jurkat cells, which are a type of T cell, part of the immune system.
And HMGCR is involved in?
Cholesterol synthesis.
Yeah.
Which is really important for how T cells function. So again, the effect lines up with the known role of the gene in that specific cell type.
So the impact of changing a gene isn't fixed. It really depends on the cellular environment it's in.
Precisely. It highlights that a gene's role and the consequence of perturbing it can be very context-dependent.
That's crucial when thinking about diseases that affect different tissues. What about the amount of change? Does it make a difference if you just slightly reduce a gene's activity versus knocking it out almost completely? Dosage sensitivity?
Yeah, dosage sensitivity. And Trey gave some really neat insights there, too. For some genes, they found the effect wasn't just like a simple linear increase.
Meaning double the change doesn't mean double the effect.
Exactly. For genes like SOX9 or parts of something called the polycomb complex, small reductions in their levels had, pretty minimal impact across the transcriptome.
Okay.
But larger reductions led to disproportionately huge changes across the whole gene network.
Like a tipping point.
Sort of like a tipping point, yeah. Once you cross a certain threshold of reduction, it triggers a much more widespread response.
That's important for therapies, right? You might need to hit a target hard enough to see an effect, but maybe hitting it too hard triggers way more than you intended.
That's the potential implication. And they even looked at how the pattern of gene changes correlated at different levels of perturbation.
Oh, interesting. So not just the overall impact, but which specific genes were affected.
Right. For some genes, like one called BCR, the set of affected genes was pretty similar whether the perturbation was weak or strong. The pattern was consistent.
Okay.
But for others, like SOX9 again and GATA1, weak and strong perturbations actually led to qualitatively different sets of genes changing.
Wow. So it's not just a dimmer switch. Changing the level can actually change what gets affected.
Exactly, which really emphasizes that if you only study a gene at one level of knockdown, you might get an incomplete picture, maybe even a misleading one, of all its functions.
Fascinating. Now, you mentioned they also applied trade outside the lab context to data from neuropsychiatric disorders. How does that work?
Yeah, that was a really interesting application. They took existing gene expression data from postmortem brain tissue of people who had different psychiatric conditions.
Okay, like autism, schizophrenia.
Exactly. Conditions like autism, schizophrenia, bipolar disorder. And they reanalyzed this existing data using trade.
And what did trade show that maybe standard methods didn't?
Well, by accounting for the noise and looking transcriptome-wide, trade gave a more robust estimate of how similar the gene expression changes were between these different disorders.
More similar than previously thought.
Yes. Trade actually suggested a greater degree of overlap in the underlying biological disruptions across these different conditions than earlier analyses had shown.
So potentially pointing towards shared molecular pathways going wrong in seemingly different disorders.
That's the suggestion. It hints that maybe some common biological mechanisms could be at play, which down the line could point towards shared therapeutic strategies. It's still early days, but it shows the power of looking at the whole picture.
Yeah, revealing subtle but maybe more consistent patterns. The paper also mentioned this metric, πDEG, the effective number of the EGs. What does that add?
Right. DEG. It's another metric estimated by trade. It basically helps you quantify how the impact is spread out.
Meaning?
Meaning, does the perturbation cause really big changes in just a few genes, or does it cause smaller changes but across many, many genes?
Focus versus widespread impact.
Exactly. And they found the median PEG for a typical gene perturbation was around 45 again, suggesting effects tend to be relatively broad.
Consistent with the earlier number.
Yep. But for those essential genes, the DEG jumped to over 500.
Reinforcing that idea of a massive diffuse impact when you hit a critical node.
Precisely. It helps distinguish between a sort of targeted molecular hit and a more systemic disruption across the cell.
That's a useful distinction. And finally, they did some gene set enrichment analyses with trade. What did those show?
Right. Two main types. First, perturbation impact enrichment. This looks for groups or sets of genes that, when you perturb them, tend to have a bigger overall impact, a higher TI score.
Okay, which genes have the biggest ripple effects?
Basically, yes. And they found that genes known to be highly constrained in humans, meaning they don't tolerate mutations well, suggesting they're important, and essential genes, those groups tended to have a higher impact when perturbed. Makes sense, right? Important genes cause bigger waves.
Yeah, that tracks. What was the second type?
Perturbation response enrichment. This flips it around and asks, which sets of genes are particularly sensitive to being affected when other genes are perturbed?
Which genes are easily knocked off balance?
Kind of, yeah. And interestingly, they found that genes that are already expressed at high levels tended to be very responsive to perturbations happening elsewhere.
So the busy genes are also the ones most likely to feel the effects of disruptions nearby.
That seems to be the pattern, yes. It really paints this picture of a highly interconnected, dynamic, and sensitive system within the cell.
It really does. So, wrapping up, the real takeaway here seems to be the value of looking beyond just the most obvious changes.
Absolutely. Methods like trade, by embracing the complexity and looking at the entire transcriptome, give us a much richer, more comprehensive understanding of how these genetic changes actually play out in the cell.
We get insights into cell type differences, this nonlinear dosage sensitivity, even potential links between complex diseases.
Exactly. It's about seeing the whole forest, not just the few trees that fell the hardest. It provides a much more nuanced view of cellular function and disruption.
This has been a truly fascinating deep dive.
Yeah.
It's amazing how these new analytical tools are letting us peel back layers of complexity we couldn't really grasp before.
It's an exciting time. By really digging into this kind of genome-wide data, we're starting to appreciate the intricate interconnectedness of life in a much deeper way.
As always, don't forget to like the video and subscribe if you haven't joined us yet.